In this video, we're going to look at how the cell copies, that is, replicates its DNA. All cells need to maintain their genetic information, that is, their DNA, between one generation and the next. For the cell, it's critical that it accurately copies the DNA, otherwise it will introduce mutations that could be detrimental to the function of the cell and may lead to diseases such as cancer. The process of copying DNA is fairly straightforward. First, the DNA has to be separated into its two strands, as each strand will act as a template in the replication process. The DNA is then copied. The replication process is described as semi-conservative, in that each daughter DNA strand will have one strand from the parent, the original DNA. DNA has a complementary base pairing system, and that A pairs with T and G with C. So this means that when we are copying the DNA from one parent strand, we are making an exact copy of the other parent strand. In the copying process, the two DNA strands are separated by an enzyme complex called DNA helicase. The enzyme uses ATP to assist the unzipping of the DNA helix. The unzipped DNA helix is then stabilized by single-stranded binding proteins, which help protect the DNA from damage and from being repaired by the cell. Now, DNA has an orientation. That is, it has a 5' end and a 3' end. Traditionally, we always write the 5' end of DNA on the left and the 3' end of DNA on the right. When DNA is copied, an enzyme called DNA polymerase adds the correct deoxyribonucleoside triphosphate to the 3' end of the growing DNA chain. That is, if on the parent strand there is an A, the polymerase will add a T to the growing 3' end of the daughter strand. If there is a G in the parent strand, then a C will be added, and so on. As the new nucleotides are added, the chain grows from the 5' to the 3' direction. However, the DNA polymerase is not perfect and it makes mistakes. And these mistakes are corrected by the proofreading exonuclease function of the enzyme, which enables it to cut out the error and replace it with the correct base. Now, as I said earlier, the DNA is unzipped for replication, and the point at which the unzipping occurs in the DNA is called the DNA replication fork, which is typically shown in figures as a Y-shaped. However, in the cell, the replication fork is actually folded back and is not Y-shaped. At the replication fork, the polymerase is held in place by sliding clamp proteins, so it can copy the DNA. Well, I can see a number of problems with what I described earlier. The problems I can see, and in no particular order, are accessing the DNA. How will the replication machinery gain access to the DNA? The DNA is tightly packaged. Plus, what happens when you unwind the DNA? How do you start to make a copy? As I said earlier, you need a 3' end to add the bases. Where does the first 3' end come from? What about speed? How long will it take to copy all the DNA in a human cell? DNA has two strands. As I said earlier, the polymerase can only add bases to the 3' end, yet one of the strands is not available in the correct orientation for this process to occur. How is this solved? Okay, so let's have a look at these problems. The first problem is gaining access to the DNA. The DNA is tightly packed, so the DNA must be unpacked to the DNA strand level so the replication machinery can gain access to the sequence. The last step of the process is the DNA unwinding from the histones, so the DNA strand can be passed through the helicase to be split and then copied. As soon as the DNA is copied, the nucleosome, the DNA histone complex, reforms and the DNA starts to repack. Now, the next problem is how do you start the process of copying? You have the DNA unpacked and the strands split. The polymerase needs a starting point, as it can only add a base to a 3' end of another base. So we need a few bases on the DNA to start the process, and these bases are called a primer. To solve this problem, the cell uses an enzyme, which is a member of the DNA polymerase family and has DNA primase activity. This enzyme binds to the DNA and builds an RNA primer of about 20 nucleotides before being swapped out for a member of the polymerase family that can add bases at a faster rate. In terms of adding nucleotides to the unpacked DNA molecule, it is fast, as the polymerase can add between 500 and 1,000 nucleotides per second. But even at that speed, the DNA in a human cell would still take between 1,600 and 3,200 hours to copy, or between 67 and 133 days. And clearly it doesn't take that long. To give you some idea of the speed of this process, if you think back to our average cell that is the size of a football and contains 23 kilometers of DNA, this means the polymerase on that scale would only be moving along the DNA molecule at around 7 to 14 meters per hour. Clearly, it doesn't take that long to copy the DNA in a cell. If it did, then pregnancy wouldn't be 9 months, but would be around 5,000 days or 16 years. So what is going on? Well, each chromosome is copied at the same time, so this reduces the time of copying to a 46th of what it would normally take, or about 3 days. But that's still a long time. So to speed things up, the chromosome has multiple origins of replication, the start points of replication, which means there is not just one site of replication per chromosome, but multiple sites. Hence, this adds more speed to the copying process. Now, there is a problem in this process, and that is the winding problem. You'll remember DNA is a double helix, so therefore it needs to be unwound to make a new copy of the DNA. Imagine the thread on this bolt is the DNA helix, and this nut here is the helicase. As you can see, the only way you can pass the bolt through the nut is either by spinning the nut or by turning the bolt. So which one happens in the cell? Well, in the cell, the nut, the helicase, stays still and the bolt will pass through it. So this means the DNA must be doing the rotating. And DNA is a big long molecule, so it would seem rather odd to have such a large molecule rotating in the cell. And I can show this with these skipping ropes that I have twisted together. On this scale, the DNA helicase would be about 14 centimetres, or about the size of this ball. So this sort of gives you the idea of the scales we're talking about. However, in real life, the helicase would be about 23 nanometres across, which is roughly 4 times the size of an average protein. Now, watch what happens when I let go of the ends of these twisted skipping ropes. They spin. And somehow we have to overcome this twisting event that is either transmitted back up the DNA strand or would cause the ends to rotate. Now in the cell, it wouldn't be the free ends that are rotating like we just saw with the skipping rope. It would in fact be the DNA upstream of the helicase. That is the DNA behind the replication fork. And we can do some quick maths to work out how quickly this would be rotating. We know that the polymerase is moving along at 500 to 1,000 bases per second, and each turn of the helix is roughly 10 bases. Therefore, this will be rotating at 50 to 100 times per second. Or if we put that into revs per minute, that's about 3,000 to 6,000 revs per minute. And if you think the average car does about 3,000 revolutions per minute at 60 miles an hour, that's quite fast. Hence, this rotation of around 50 revolutions per second would cause significant strain and could cause this DNA helix to break. Now, the cell has a simple solution to this problem. It uses an enzyme called DNA topoisomerase to reversibly nick and cut one of the strands. And this produces a rotation point close to the helicase. So instead of getting the transmission of the forces up the strand, we actually get it spinning just behind the helicase, and that is enough to solve the problem. Now, the final problem that we have identified is the strands. DNA is anti-parallel, which means one strand is going left to right 5' to 3', whereas the other strand is going left to right 3' to 5'. As DNA can only grow in the 5' to 3' direction, that is, the nucleotides can only be added to the 3' end of the growing chain, this means DNA can be easily copied on one of the strands as the template DNA is presented to the polymerase in the right orientation. whereas the other strand can only be copied in short stretches of 500 to 2000 bases as they emerge from the helicase. Think about it. As the strands unwind, one strand is presented with the 3' end to the polymerase and therefore can be easily copied once the DNA primase has added the RNA primer. And the polymerase can start adding bases to the 3' end. The helicase then presents a constant flow of available template 3' end first. We call this strand the leading strand. The other strand, which we call the lagging strand, first presents a 5' end, and the system has to wait until enough of that strand appears from the helicase so the primase can add the primer and copying can begin. However, the polymerase will soon run out of template and has to wait for more to appear from the helicase and for the primase to add another primer. Hence, on the lagging strand, the DNA is produced as short stretches of 500 to 2000 bases. And these short fragments are called Okazaki fragments, and these fragments will eventually be stick together by an enzyme called DNA ligase to make the full DNA molecule. You might think that the cell could get around this problem by waiting until all the strand becomes available. However, the cell can't wait for all the DNA on the lagging strand to be unpaired, because if it did, the presence of that much single-stranded DNA would trigger the DNA repair systems in the cell. Again, this is one of these things that appears and sounds very complicated, but a few chocolate hobnobs, a really hot cup of tea, and some time spent drawing it out, and you will soon see how it works. Okay, so in this video, we've looked at DNA replication, and we've seen how it is fast and yet slow, and how the cell makes up for the apparent slowness by copying all the chromosomes at once, and having multiple copy sites working per chromosome. We've also explored the unpacking and the winding problems, and talked about leading and lagging strands.